And welcome back on this is now the third presentation, which will finish off what we started yesterday afternoon on biomass composition. Yesterday we talked mostly about sugars. We talked about sugars and lignin, polymers of sugars like cellulose, hemicellulose, and let's see, starches, amylose and amylopectin. And today we're going to go further on proteins, etc. We're going to start off with proteins, but we'll round off the composition um, this morning. And then, of course, there's the homework associated to this, which has a research question attached to it. So, what are proteins? I think most of you would know approximately what a protein is, maybe even more than approximately, but just to get everybody up to speed. Proteins are chains of 20 different types of amino acids. Not always true, actually. Well, there are 20 found in every living cell. Well, that's not, actually also not true. But 20 proteins are coded by plant DNA. Uh, some are missing a few. Those are called essential amino acids. But there are also amino acids which are coded in mammals, specifically selenocysteine, which is not found in plants. So there are 21 amino acids, but 20 are what we call like the basic ones, the ones that are in everything. They are joined together in any linear order. So if you denature a protein, if you make it flat, um, it's linear. So the one protein is chained to the other protein. However, um, that is the primary structure. So the sequence of amino acids is its primary structure, so as a string and an alphabet of 20 different symbols. But it has a secondary structure because it folds and crumples, yeah, into a massive shape. And online, if you go to my YouTube channel, I have a couple of amino acids, and also you can find 3D models of proteins like BSA, which bovine serum albumin. It's a complex three-dimensional structure as a secondary structure. Then you have a tertiary structure where these secondary structures are then attached to. So if we look at our hair, for example, right? If you go to the keratin in the primary structure is a string of amino acids, which then bundle together to form a keratin uh, uh, molecule. They bound together into different sequences and structures, and that's why your hair, the one hair is slightly different than the other hair, and on that, in my hair is different than your hair, but not only that, on one strand of hair, this sequence is different than this sequence. So it makes working with proteins a little bit complicated. But and proteins can range from just a few amino acids to thousands and thousands. Here, as I mentioned, are the 20 amino acids which we consider to be the normal amino acids found in all life forms. They can be grouped into different subsections. We have the unpolar hydrophobic ones. We have the polar and the neutral ones. We have this is in German, sauer. So we have the acidic, and we have the basic. And this is a simple way of breaking them down. Actually, my research is on amino acids. I break them down slightly differently. I break them down their structures. But this is a simple way of thinking about them. So we have two negatively charged side chains, three positively charged, and these two are mostly uncharged, but not completely uncharged. Some of them are uncharged, but some of them still have charges, but less than these five. And some of them are hydrophobic, and some of them are polar, so they dissolve quite well in water. Yeah. I'm not sure how much I could literally speak hours and hours on amino acids, but I'm not sure how much you guys would be interested in me talking about my research. Oh no, are there connection issues? Yeah, that happens sometimes. The server the server that we are on will kick you off um, because of what's it called, maintenance. But you normally can just click on reconnect and it brings you right back. I'm sorry if it happens to you. Normally it'll happen just one time, and but just continue. I'm 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 recording this anyway. You get kicked all the time. You might want to check your firewall settings. Okay. At the end of at the end, I'll just continue. Yeah, if you're not using Chrome, I highly suggest. Okay. All right. I'll continue. Hopefully, this is a just a minor glitch. Now that's what amino acids are. So I'll continue. Hopefully, the small server errors have been taken care of. Proteins' role in life. So the structural proteins are like organisms' basic building blocks. So collagen, nails, hair. Proteins give structure to your cells. They're everywhere. In enzymes are proteins. They're very small proteins normally, just a few amino acids long. They're the biological engines which mediate a multitude of biological reactions. Most importantly, ribisco in leaves. Then you have, like I mentioned, transmembrane proteins. These are the housekeeper cells, and the housekeepers of the cell. They regulate the cell volume, the extraction. They do transport across the cell wall. They they concentrate small molecules and the extracellular environment outside. They generate ionic gradients, which are essential for muscle and nerve cell function. So the sodium-potassium pump, ding, 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 please remember that, is an example of this transmembrane process. So just a quick link to the project. This is exactly why Grasa, Johan Sanders von Grasa wants you to look at potassium, because the sodium-potassium pump is an example of this membrane transport. And then of course you have storage proteins. So seeds, you probably know, have a lot of protein. Protein is like fat, is a very good storage molecule. Recovering protein from biomass. So biorefining is not just for producing biofuels. A lot of people think biorefinery biofuels. That's not the case. For economic feasibility, you must also focus on protein recovery as an essential part of, like, a first step. Because if you can extract the proteins, there's still all of the lignin and the cellulose and the hemicellulose that you can use for fuel. The protein is really too valuable to waste. Several biomass sources are preferred. Protein hydrolysis of amino acids. So, as I mentioned, proteins are made up of amino acids. You can actually break the connection between the amino acids, and this is actually a large part of my research. My most of research is on proteins, amino acids, but the hydrolysis of amino acids. It's the new line people are focusing on. Selective recovery through crystallization. Again, I didn't make this slide, but this person's brilliant, because this is exactly what I believe. And in the bigger picture, with other components. So proteins, amino acids, crystallization, and as a beginning step before making biofuels. Right, so here's the protein biorefinery. You can extract the biomass. We're going to go into more detail at the next two presentations on extraction techniques. But right now I'll just name them. You have alkali and enzymatic extractions. You can, of course, screen them. You have T residues, Jatropha tree. These are different sources of the biomass, and the co-production of oils and sugars. So you have seeds that you have. You press seeds for oil, like peanut oil or any kind of seed oil. And what's left over is a press cake, and this press cake has a lot of proteins in it because seeds are mostly oil, but they're also high in proteins. So that's another good source. Once you have the protein, either you can crystallize it or extract it directly for protein applications of feed or fuel. You can also use enzymes or chemicals to hydrolyze the proteins to amino acids if you don't stop with just normal proteins. The amino acids themselves, a combination of amino acids is worth something. However, you can separate them using membranes, chromatography, crystallization, and solvents to make individual amino acids. And again, we're going to talk about protein recovery in the next couple of lectures. But biomass is the big part. It can be separated between proteins and lignocellulose. The proteins can then be divided into amino acids, and then amino acids can be extracted individually. And each step brings more added value. Yeah, so you're going further and further up the bio-based pyramid. But it also costs a lot of money to do this. Chromatography costs millions of euros. Large columns cost a lot, and that's just to run them. It costs millions and millions. Okay, so protein recovery, you have this hydrolysis into amino acids and then separate them. And we separate them by their properties. So I mentioned you have acid ones, base ones, hydrophilic and hydrophobic. You can separate them by, for example, distillation using ethanol or hydrolysis to amino acids. Because some amino acids, if they are very hydrophilic, if you add ethanol, they are no longer dissolvable in water because they are not. They like water, they're hydrophilic, and they don't like ethanol. So if you add ethanol, not only proteins but also some amino acids will precipitate or crystallize. Now, protein content. Yesterday I showed you for a few what their gross content was. Now specifically for proteins, you can see here are lots of different biomasses. So here's spirulina, which is approximately 65% protein by dry mass. Poultry feather is about 90. I've measured 95%. Different grasses. So this is switchgrass, has about 9%. Sugar beet leaves have about 24%. And we're very interested in seeing what you guys can extract, how much proteins you can extract. One problem, you cannot use these this data as for your design. Why? Because this is total protein, and so let's say, for example, sugar beet leaves. It says about 23% proteins, but this is for everything, including the proteins within the cell wall. And if you mechanically, we're going to use the hand blender, if you mechanically release proteins from the cell, you will not extract proteins in the cell wall. They are stuck, remember, in that three-dimensional lignin cage. And lignin is very resistant to especially mechanical extraction techniques. You're really not going to get the protein out from the cell wall. You will get the proteins from the cytoplasm, from the nucleus. So even though here it says sugar beet leaves 23% protein, you will not be recovering 23% from dry mass. Oh, let's pause. Okay. Uh, Back to presentation. So each of these biomasses, this is total. And I remember when I first started doing research on proteins from these different biomasses, I didn't get anywhere near the amount of protein, and this is why. Because you have to do, it's a combination of techniques to get to the highest percentage of the protein content by dry mass. And in our client last year, we used mechanical but also thermochemical techniques of extraction. But your client this year specifically does not want you to use thermochemical. He wants you just to use mechanical. So that's why we're not using thermochemical. And I'm just warning you now, you will not receive the results anywhere near what you see here. If you do, I'm really excited. Please tell me what you did if you get such high percentages. Okay, and again, the percent of amino acids within the protein is different. So chicken feathers are well known to have lots of cysteine, L-cysteine, which is here, L-cysteine. This is a hydrophilic protein. So it's very good to have in things like bread, so it stays fresh. Because cysteine is also in cactuses. It's a good way. It bonds with water extremely well. So cysteine is a good one. Glutamic acid is another very economically interesting amino acid. Glutamic acid is used in, for example, monosodium glutamate, which is a salt substitute, which is very popular in Asian cuisine. Aspartame is a combination. You may know aspartame. It is a sweetener. It's made from aspartic acid and phenylalanine, on top of my head, I believe. So those two are also very marketable. If you can find a way to extract aspartic acid or glutamic acid from proteins, you're doing quite well. And by the way, that's what I'm doing. But we'll see. The targets are for example, if you can get glutamic acid, you can make all sorts of things. So as I mentioned, monosodium glutamate, which is a good for food additive, but you can also make vinyl. You can make acrylonitrile, which is for in all sorts of things, such as your clothes and and polymer plastics that keep in large tanks of sharks. But they're also in your lab gloves. If you have I think that's blue colored one, those are the acrylonitrile gloves. People who are allergic to the normal gloves. You can make nylon and other methylpurolidine products. So if you first can find glutamic acid as a product from the biorefinery, it has lots of applications. A lipid, a fat, is a compound that is insoluble in water but soluble in an organic solvent. Organic solvents we'll be going into again more, but ether, benzene, ethanol, acetone, chloroform, these are all organic solvents. Lipid is synonymous with fat, but also includes phospholipids, sterols, etc. The chemical structure of lipids are glycerol plus a fatty acid. Lipids are concentrated sources of energies. So a lipid has 9.45 kilocalories per gram, which is more than double of sugar or protein. They are both around 4 kilocalories per gram. So lipids are a very good storage of energy. The structural element is of a cell. lipids are. There's subcellular components involved also. They are also storage in seed. So maybe you remember, some of you were taking cell biology, right? And I think some frowny faces for people retaking cell biology. I'm sure then know that there's a phospholipid bilayer in cells. So there's lots of lipids in cells. Here is what the basics of a fat cell. You have a glycerol plus a fatty acid. The H's combine, so you and an O, which so you these bind here, releasing 3 water molecules, and here you get a triglyceride lipid. We have different classes of lipids. A simple is can be esterified with glycerol, so it's a fatty acid esterified with glycerol. So like here, you have a fatty acid and a glycerol. That's a simple. You have compound, which is the same as a simple, but with other compounds also attached. So they just have maybe a phenol, maybe whatever attached to it. A phospholipid is a fat which contains phosphoric acid and nitrogen. Example is lecithin. You have glycolipids, which are fatty acids compounded with a CHO, a kind of sugar. A derived lipid is a substance from the above derived by hydrolysis. So if you hydrolyze and it becomes, it's transformed, it's a derived lipid. And sterols, large molecule weight alcohols found in nature which can combine with fatty acids. An example of this is cholesterol. So cholesterol is basically a combination of fatty acid with an alcohol. We also then combine them in between saturated versus unsaturated fatty acids. A saturated fatty acid, probably you've run across this before in looking at what foods to eat. The saturated fatty acid of a lipid have no double bonds between carbons in the chain. They're all single bonds. A polyunsaturated has more than one double bond in the chain. Yeah, so it's not saturated with hydrogen. If it's completely surrounded by hydrogen, then it's saturated. Yeah, you can think of it like that. When hydrogen's in water, you can think about. In Dutch, it's called waterstof, so you can even think about it like that. It's saturated and unsaturated. Some hydrogen molecules are missing, let's say, and it has a double bond between the carbon because there's an electron there. It has to bond somewhere, so it double bonds with the other carbon, and that's why it's unsaturated. It's missing a connection, so it doubles up on a carbon. Most common polyunsaturated fats, or PUFAs, contain the polyunsaturated fatty acids oleic, linoleic, and linolenic. Sorry, it's difficult to say. Unsaturated fats have lower melting points. Yeah, so if it's unsaturated, it has a low melting point. Can anybody tell me, give me a fat that they know which is liquid at room temperature? For example, something you might cook with. Huh? Yeah, olive oil, cooking oil. Just so that's why we say here plant fats have a much higher PUFA, and rapeseed, sunflower oil, exactly. All correct, 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 correct. And what do they all have in common? They're all plants. Well, that's true. Most, not all, some of them, it's still lower, let's say. But if it has a polyunsaturated fatty acid, so it has a sorry, if it's stearic, it melts at 70, oleic at 26. And plant fats are much higher in this polyunsaturated fatty acids. Therefore, exactly. Well, 25 is, yeah, 25 is still pretty it's pretty straight low compared to, who can name me a fat that we use for cooking, for example, which is at room temperature solid? Lard, yeah, lard. And what else? It's like my favorite thing in the world. Butter, yeah, Laura's on fire this morning, Laura. Exactly. So animal fats like butter, like that's churned milk basically. It's the fat of milk. So those fats are solid at room temperature and therefore have a higher melting point. You should keep this in mind when you think of extraction techniques, right? So if you want to extract fat from a liquid, you just have to cool it off. And you see that too if you make like chicken soup. When you boil the chicken soup, it's completely clear, and then when it cools down, you might see a film of fat on top of the chicken soup. That's because at a high temperature, it melts in water, it dissolves in water, and at a lower temperature when it cools off, if you put it in the refrigerator, for example, it will harden. Okay, which another reason why you don't uh, you don't use certain oils to bake to fry. Anyway, so fatty acids commonly found in lipids, butyric, palmitic, stearic, are saturated. Oleic, linoleic, and linolenic. Melting points of the unsaturated fatty acids uh, are, it says liquid. That's for palmitic and stearic, it's around 63 or 70. And for the others, it's liquid at room temperature. The only saturated fatty acid in this list, which is liquid at room temperature, is butyric. Yes, exactly. Thank you, Yannick. Are you still getting kicked out, by the way, or is that stopped? Oh, okay. Yeah, it goes through a cycle. Like I said, they restart the. Sorry for all the confusion, but yeah, it's annoying. Fatty acid nomenclature. Nomenclature reflects location of double bonds. Again, also used are common names. Linoleic is also known as 18,2 and 6. That means it has 18 carbons in length, 2 double bonds, the first of which is on the 6th carbon. Arachidonic is 24 and 6. So what does that mean? 20 carbons long, 4 double bonds, of which the first is on the 6th carbon. Yeah, 4 double bonds. Larry, you're on fire today. This is really your subject matter. Sometimes some things you just click. All right, so here's an overview of the different fatty acids and where you can find them. You can use that for your research. Now we're going to go to nutrients. We have phosphorus, magnesium, calcium, potassium, and nitrogen. They are distributed through a species. There are substantial differences between crops, plant material, and minerals. And they are present in what we call the ash fraction. So ash, oh no, the ash fraction is the extract. Uh-oh, pause. Uh-oh. Okay, that was a quick break. Now we'll continue with the ash fraction. So if you asked at the very beginning, there were like 6 or 7. I forgot the components. I think 7 components. One of which was ash. Ash is what we call is the nutrients that the other amount. of the biomass that we are not involved in biomolecules. So this is nitrogen, um, phosphorus, magnesium, calcium, potassium. Again, this is in German. Uh, so this is lucerne grass. This is straw, uh, seed, and root of wheat. This is the planty part of the potato, and this is the root of the potato. And here we have of the green leaves, we have the leaf. Huh? Yeah, yeah. Okay, so now we can fill in at the very, 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 very beginning of yesterday's class. Remember, on the first sheet, I had this empty section. Now we can fill it in, and hopefully you will also fill this in your project. So if you have leaves from corn, cassava, here's spirulina, algae, the seed of the rubber tree, wood, straw, sugar beet. Uh, I don't think this is leaves. I think this is the total sugar beet, and also the grain of the wheat. We can fill that in. So I'll take spirulina since one of our groups has that. It's 80 to 90% water, 0% cellulose, 30% hemicellulose, 0% lignin, half is protein. So now you know why spirulina, dried crushed spirulina is used as a protein supplement. 5% fat. So, and fat you can normally get rid of by just lowering the temperature. So if you freeze dry it, the fat will float away. Ash is 14%, and this is basically nutrients. So spirulina is an excellent choice for protein extraction. However, these proteins, they're not all proteins are the same. Hopefully you remember that. So there are the green and the white proteins, and they're each worth different amounts. Okay, no coffee break. We've already had a couple of breaks. I just want to finish this so you guys can go and do your homeworks. I think most people would agree. And here's the final products you can use them for. So salts for fertilizer, fat and oils for energy food, feed for protein, food for starches, energy for lignin, a food additive for hemicellulose, and paper from cellulose. Building blocks you can fill in also here for each product. And now it's filled in together. So if you have cellulose matter, building block is glucose, the pretreatment is hydrolysis, conversion through fermentation, you can distill it into ethanol. Yeah, and these are the different processes that you can use, for example, from proteins. You can get bulk chemicals, nylon, through crystallization, which you get from hydrolysis and extraction of amino acids. I'll let you go back to that. It can be extremely complicated. Obviously, you don't need to make all of this. You need to look at just one specifically. Proteins. Recommended products. If you want to learn more, you can read this article, Renewable Resources for the Bulk Chemical Industry. It's an excellent article written by some friends of mine, but it's extremely good. It's a little bit outdated. It's I think 2005 it was written, but it's quite good. It's dense too. It took me a long time to get that out. But if you're interested in amino acids and what you can make from it, you definitely want to look this article up in Explora. Names of the refinery are based on their origin, talking about that in the next lecture. The energy or product given, the conversion technology, so is it biochemical, thermal, chemical, or mechanical, and the platform, which is the product, so protein, let's say. And this is I'll let you use this for your research. I don't want to talk about every single step, but this is a very good way of looking. If you have like a grass, what you can do with it. Use this when you're doing your biorefinery design in your project. It's obviously it's way too much to discuss. We could spend an entire class just talking about this one sheet, but I don't want to do that. That's too much detail. But use this as a tool for in your project. So if you have grasses, you can fractionate it to different parts. So just follow the arrows which you can make from it, and let that inspire you. So you have grasses, but you might also be looking at the lignocellulose also. If you have something that's strong in lignocellulose, you can go through different pathways. All right, tomorrow, which is actually this afternoon. Last year this was split differently. So this afternoon we'll be talking about biorefinery types. Thank you for your attention. I will upload this video to Blackboard. I apologize for the interruptions. In my experience, the getting kicked out happens intensely, but then it stops. And I will be here on my computer and physically in LA 008 until 1 o'clock if you have any questions. Thank you.